السلام عليكم ورحمة الله وبركاته. Transcription. في البداية هنتعرف على types of RNA in the cell. Types of RNA. Number 1, messenger RNA. Forms about 5% of RNA in the cell. The most heterogeneous type of RNA. Each protein has a specific mRNA responsible for its synthesis. Transfer RNA. Transfer RNA forms about 15% of the cell of RNA in the cell. Clover leaf shape زي ما احنا شايفين. It has 4 arms and a variable extra arm. It has 4 arms with extensive base pairing زي ما موجود في الصورة كده plus a variable extra arm. 4 unusual nucleotides are present in the tRNA. ال tRNA بيحتوي على 4 unusual nucleotides اللي هم thymidine, pseudouridine, dihydrouridine and inosine. Function of tRNA. Carry of amino acid in the process of translation يعني act as adapter molecule. أثناء الترانسليشن وأنا بصنع البروتين مولكيول carry الأمينو أسيد وبالتالي act as adapter molecule بيعمل adaptation ما بين البروبر كودون مع البروبر أمينو أسيد. دي صورة ال tRNA. It has acceptor arm اللي هو ده. الacceptor arm ends in a specific sequence اللي هو CCA sequence. The 3' end of the sequence ده carry الأمينو أسيد. يبقى أنا عندي the acceptor arm end in CCA sequence and the 3' end of the acceptor arm accept the amino acid. The anticodon arm end in a loop that contains 7 bases. The 3 bases in the middle constitute the anticodon. الانتيكودون arm بينتهي بلوب. اللوب ده بيتكون من 7 bases. 3 منهم بتمثل الانتيكودون arm الانتيكودون اللي بيتعرف على الكودون. Ribosomal RNA اللي هو ال rRNA form 80% of the RNA in the cell. Constitute about half of the ribosome. بتمثل حوالي 50% of the ribosome. Small nuclear RNA ده النوع الرابع من ال RNA. Small nuclear RNA are found in the nucleus. Small nuclear RNAs are associated with protein forming small nuclear RNAs اللي بنسميها باختصارها SNRP, which are responsible for splicing of mRNA. Transcription. The definition of transcription is the process of RNA synthesis from DNA. The enzyme responsible for transcription is called RNA polymerase enzyme. Only one strand of the DNA is transcribed وبنسميه في الحالة دي template strand أو antisense strand أو non-coding strand. This strand vary from gene to gene. يعني مش strand واحد ثابت كtemplate لكل الجينز. RNA polymerase reads the template strand in 3' to 5' direction only. يعني ال RNA polymerase هيقرأ الtemplate strand in 3' to 5' direction only في اتجاه واحد فقط. وبالتالي build ال RNA in 5' to 3' direction. This complementary RNA is called mRNA أو messenger RNA. ال RNA اللي بيتصنع في الحالة دي اسمه messenger RNA وبيتصنع complementary للtemplate DNA strand. The other DNA strand بنسميه sense strand أو coding strand which have the same sequence as the transcribed RNA. ال other strand اللي هو non-template strand بنسميه sense أو coding strand لأنه بيبقى شبه المسنجر RNA اللي أنا صنعته لأن هما الاتنين complementary للtemplate strand. Eukaryotes have multiple RNA polymerases. RNA polymerase 1 produce 45S rRNA. RNA polymerase 2 produce mRNA plus small nuclear RNA. RNA polymerase 3 produce tRNA plus some small nuclear RNA plus 5S rRNA. Chromatin remodeling. Chromatin present in the cell in 2 forms, eochromatin and heterochromatin. Eochromatin the actively transcribed gene and found in a relatively relaxed form of chromatin. But heterochromatin, this is inactive form. chromatin remodeling المقصود بيها dynamic changes in the chromatin structure between euchromatin and heterochromatin forms. Chromatin remodeling is achieved by histone acetylation and deacetylation. Conversion of inactive gene to active form achieved by histone acetylation by acetyl transferase enzyme, while deacetylation by deacetylase enzyme lead to inactivation of the gene. So, acetyl transferase enzyme produce acetylation to histone and so convert inactive gene to active gene. This allows transcription factor to act on the DNA and increase transcription. But deacetylation leads to decreased transcription. Transcription in eukaryotes have 3 steps: chain initiation, chain elongation, and chain termination. Chain initiation. RNA synthesis is initiated at a specific site on the DNA template strand known as promoter region. RNA polymerase enzyme is responsible for recognizing the promoter region, make a complementary RNA strand, recognize the termination region. Eukaryote genes contain promoter sequence. That's data box or cat box. Data box is known as Hogness box. In other gene constitutive genes in the cell, GC box is present instead. Eukaryote genes contain specific sequence known as enhancer and silencer sequences. Also, they need transcription factors. This is protein that help to recognize the promoter region. Chain elongation. A portion of DNA template strand unwind at the point of RNA synthesis. This forms a short length of RNA-DNA hybrid. The unpaired bubble of DNA in the open initiation complex travels along the direction of RNA polymerase. في البداية هيحصل partial unwinding and separation للدبل strand DNA at the point of RNA synthesis. مش كل الجين هيفتح مرة واحدة، جزء بجزء. RNA polymerase enzyme هيمسك مع التمبلت strand of the DNA ويبتدي يصنع لي complementary RNA strand اللي هتكون في البداية عندي ماسكة مع ال DNA في صورة RNA-DNA hybrid. الأكتف site of transcription بنسميه transcription bubble. يبقى this active site of transcription بنسميها transcription bubble. هيبتدي ال RNA اللي أنا صنعته يحصل له separation من ال DNA RNA hybrid، وال DNA بعد اللي هو باين في الصورة هنا، في صورة هنا ستراند لونها أحمر، هيبتدي ال RNA يحصل له separation، هيبتدي DNA double strand يحصل له annealing تاني behind ال RNA polymerase ويفتح in front of ال RNA polymerase، ودي بنسميها traveling أو بنوصفها traveling of the transcription bubble throughout the gene along the DNA gene. Chain termination. Chain termination is very poorly understood. However, it appears that the termination signal exists far downstream of the coding sequence. Inhibitors of RNA polymerase 2. Alpha amanitin. This is a potent toxin from specific mushroom. It blocks the translocation of RNA polymerase during transcription, وبالتالي inhibiting transcription. It forms a tight complex with RNA polymerase enzyme, thus inhibiting messenger RNA synthesis. Mitochondrial RNA polymerase. Mitochondria contain a single RNA polymerase that's responsible for synthesis of mitochondrial protein. It resembles bacterial RNA polymerases. Action of antibiotics. Number 1, rifampicin or rifampicin, used in treatment of tuberculosis. Rifampicin interfere with the formation of the first phosphodiester bond in the RNA chain. على مستوى الإيوكاريوت RNA polymerase does not bind to rifampicin and so RNA synthesis is unaffected. Actinomycin D, the first antibiotic to have therapeutic application in tumor chemotherapy. Actinomycin D bind to the DNA template and interfere with the movement of RNA polymerase enzyme along the DNA. ده على مستوى both الإيوكاريوت and البروكاريوت. Both transcriptional modifications and اللي بنسميها RNA processing. In prokaryotes, messenger RNA is ready to synthesize the protein. Once إن هو اتصنع ready for protein synthesis. على مستوى الإيوكاريوتس the primary RNA is called heterogeneous nuclear RNA. heterogeneous nuclear RNA. heterogeneous nuclear RNA is modified into mature messenger RNA. يبقى المسنجر RNA primary molecule of messenger RNA need multiple modifications to get mature. The mature messenger RNA then moves out of the nucleus into the cytoplasm for translation. هنتكلم دلوقتي على الإيوكاريوتيك messenger RNA modifications اللي بنسميها RNA processing. Number 1 capping. Capping كدefinition addition of methylated guanine nucleotide at 5' end of messenger RNA. 7-methyl guanine is added by unusual 5' to 5' triphosphate linkage. The function of capping, the addition of 7-methyl guanine. Number 1 to protect the messenger RNA from 5' X nuclease to enhance translation of messenger RNA. تاني modification اللي هو البولي adenylation addition of poly A tail at the 3' end of messenger RNA up to 40 to 200. Function protecting messenger RNA from 3' X nuclease, facilitate their exit from the nucleus. Number 3 removal of introns أو splicing. Definition removal of introns and rejoining الإكسونز. The primary messenger RNA transcript عبارة عن coding sequence اللي بنسميها exon plus أو interrupted by non-coding sequence اللي اسمها الانترونز. Splicing here we remove الانترونز and rejoining الإكسونز again to make messenger RNA active for translation. Spliceosome is responsible for splicing. الصورة دي بتوضح الجين. في البداية عندي الجين أو على مستوى الدي ان ايه الإكزونز اللي هي الكودينج سيكونس interrupted by الانترونز اللي هي النون كودينج سيكونس. مع الترانسكريبشن البرايمري ترانسكريبت عندي بيبقى ياخد نفس مواصفات الجين عبارة عن اكسونز بلس انترونز. عن طريق السبلايسنج يبتدي يحصل ريموفال للانترونز اللي هي نون كودينج سيكونس اند ريجويننج للاكسونز اللي هي الكودينج سيكونس تو جيت ماتيور ترانسكريبت ريدي فور ترانسليشن. A few eukaryotic primary transcript contain no introns. بعض messenger RNA primary transcript does not contain introns. For example, histone messenger RNA does not contain introns. Also في عندي حاجة اسمها alternative splicing. Alternative splicing دي give rise to different messenger RNA and hence different protein from the same initial primary transcript. Thank you.